So the reason why I'm showing you this image of Mount Wellington, you might be wondering why in the world I would want to show you an image like this is for a very good reason. So current estimates are that there is somewhere in the range of 100 billion stars in the Milky Way. And that's basically a give or take. Well, what we know for the world's oceans is that there's somewhere in the range of 5.25 trillion pieces of plastic floating in the top 10 centimeters of the ocean surface layer. So what that essentially means is there are approximately 50 times more pieces of plastic floating at the surface of the ocean than there are stars in the Milky Way. A lot of the research that I do on remote islands deals with big numbers like this. The numbers are really vast, and this is because debris is accumulating on beaches worldwide, at the bottom of the Marianas Trench, for example, even on the surface of Mars. When we're dealing with thousands of seabirds killed by ocean debris, hundreds of cetaceans entangled in plastic, trillions of pieces of plastic floating on the ocean surface, millions, billions, trillions, these are big numbers that become very difficult to comprehend. This is essentially big data. And so trying to figure out what to do with this data and how to relay this information to the public in a way that is understandable is a big part of what I do as both a scientist and a science communicator. It's trying to find analogies. So let's start at the beginning and talk about some of the things that plastic actually does. So when plastic enters the ocean, it acts like a magnet or a sponge, and it basically accumulates toxins onto the surface of the plastic so that the concentration of those chemicals on the surface of that plastic can be multiple orders of magnitude higher than the concentration of those same chemicals from the seawater in which it came. This is exemplified by the panel on the bottom. So you can basically see plastic resin pellets, which are relatively new, and so they're fairly clean looking on the left-hand side of that panel, through to resin pellets that have presumably been in the ocean for many years, possibly even decades. And the surface of those pellets have become quite degraded and abraded and has essentially become quite textured and porous and has increased the surface area. And along with that, the chemicals are able to adhere and absorb to the surface and become those elevated concentrations. These become almost like little toxic bullets. So some of the work that I've been really fortunate to be involved in involved the Australian Synchrotron Institute, which was based in Melbourne. And essentially what we did was ask the question, how much chemical is actually adhered to the surface? And is it actually true that the concentration of these chemicals is elevated on the surface? Or is it that it's actually fairly evenly distributed throughout the plastic? In other words, is it that we're dealing with chemicals that are compounded into the matrix of the plastic at the time of manufacture, for example, and so it would be fairly evenly distributed? So what these figures are showing, what you can see in these figures, is basically on the left-hand side, what we can see is a thin microtome slice through a white bottle cap. And the image in the middle is actually one of these nurdles or resin pellets, again, a thin slice through it. And the colorful panels just to the right of both of those shows the concentration of different trace elements that we were able to quantify using something called XFM. And essentially what these two things show is that the concentration of these trace elements, be it bromine, zinc, iron, or really a number of things that we looked at, is much higher on the surface of these plastic particles than it is at the core of that item. In fact, the elements are so high and there's so much kind of absorbed to the surface of some of these particles that it really threw us for a loop. So for example, in that middle panel involving the plastic resin pellet or the nurdle, we actually believed this to be a black nurdle or a black pellet. And it wasn't until we microtomed through it that we saw that the core is actually translucent or clear and realized this was a white pellet. So that pellet is so old, discolored, and degraded, and presumably has such a high concentration of various things absorbed onto the surface that it completely fooled us. So what does this mean? What does all of this really mean? And what we're most concerned about is when wildlife, for example, pictured here, a seabird, consumes these particles. A lot of this chemical is absorbed to the surface. And so when it's exposed to things like digestive enzymes in the stomach of a bird or maybe a whale or a tuna, is there capacity for some of these chemicals to be liberated and released out into that animal's tissues? This is a core question that drives a lot of the research that my lab does, and we'll be able to answer some of that over the next couple of slides. So this is the study system that I've been working in over the last decade or so. We often use seabirds as sentinel species or indicators because of their position at the top of the marine food web. They're highly reliant on the marine ecosystem for their survival, and we can make some fairly safe assumptions that, for example, if the seabird population is doing pretty well, if their breeding success is high, if the population is increasing, that the health of the surrounding ecosystem, the prey that they feed on, must also be doing well. And so we monitor one or two sentinel species really intently, and that gives us an indication of kind of the overall health of that system. And so what has started to kind of worry us is that we now know that seabirds around the globe, not just the ones here on Lord Howe Island where I work, are largely declining faster than any other bird group. And so that begs the question, why? Why are they declining? Why are they not doing very well? And could plastic possibly be playing a role? So I've been very fortunate to be involved in this project, which dates back to 2001. This is the longest running plastic ingestion data set in the Southern Hemisphere and the second longest running in the world. And this has allowed us to ask and answer certain questions that otherwise wouldn't be possible. It's allowed us to look at trends over time, also to investigate different spatial scales because we have complementary data from places like Western Australia for the exact same species. So what have we learned? Well, at a very basic level, we've learned that in a relatively short period of time, the proportion of birds that are actively ingesting plastic has gone up quite rapidly, from around 75% of the proportion of the population containing plastic in 2005 to more than 90% in 2011. So in the span of around 5 years, give or take, the population was significantly more impacted by plastic in a really quick period of time. And since 2011, the proportion of birds consuming plastic has remained relatively consistent between 90 and 100%, depending on the year. So it's kind of alarming for us going to Lord Howe Island and realizing that almost any bird you pick up almost certainly contains some plastic. What we've also found is that there's other metrics that we use to look at the impact of plastic as well, not just the proportion of birds. We also look at things like the average number of pieces of plastic per bird. And that has also been going up over time. So in the early 2000s, on average, we would find somewhere between 3 and maybe 5 pieces of plastic per bird. And now that can be upwards of around 10, 20, sometimes even 40 pieces of plastic per bird. And our record holder was a whopping 276 pieces of plastic in one 90-day-old chick. That plastic accounted for 14.9% of that bird's body mass. So if you think about that from a human perspective, that's equivalent to you or I having somewhere in the range of 10 kilos of plastic in your stomach. So those are some pretty alarming statistics. The million-dollar question is, what is it doing? I think the public perception, and fair enough, is that, well, duh, it must be doing something and it must be really, really bad. But there's actually a lot of really active debate in the plastics discipline about what this is actually doing to wild species, how frequently negative impacts occur, and whether or not this ever has the capacity to do things like drive population trends. So a lot of what we look at is what we call sublethal effects. These are harmful things that are largely invisible to the naked eye. And here again, I want to use another analogy. So think about someone in your family, maybe someone you know at school who maybe has diabetes or needs kidney dialysis treatment. Those people are suffering and their lifespan will probably be affected. But by and large, you don't know about it. You can't see it on the outside of that person. You just don't know unless they tell you. But when we're dealing with wildlife, they can't communicate these issues. They can't say, I hurt here. And so as a scientist charged with the task of trying to quantify how bad plastic is impacting species, when some, possibly most of these impacts are largely invisible, is a really significant challenge. So we've tackled it in a number of ways, and I'm going to show you just 4 of those ways. So the first one is relatively basic. We look at things like body condition, body size. So here I'm showing you some results from a paper from 2014. And we looked at, if you look at the left-hand panel, the relationship between just bird body mass and the number of ingested plastics. And you can see that there's a bit of a downward trend, a negative relationship. So the more plastic you have in your stomach, the lower your body mass. And that seems fairly intuitive. You're being fed more plastic by your adult parent and less of the good stuff, less fish, less squid. This is not good news. The same thing we observed for things like head size and bill length, so kind of overall measures of body size. Again, if you have a lot of plastic in your body, you tend to have smaller size. So to step things up a notch, we looked at this issue of chemicals. So now we know that plastic has reduced kind of body size and overall condition. Is it exposing animals to chemicals in the way that we thought it might? So what we can do with wildlife is we can test a range of tissue types, whether that be blood or in this case, feathers. And so we have a nice paired data set where we have a known amount of plastic recovered from birds, and we also sample tissues from those same individuals, and in this case, feathers. What we basically found is that feathers from this species contain quite a high level of a number of chemical contaminants. In this case, I'm showing the results for just a handful of trace elements. We've looked at other things, including some organic contaminants, PFAS and other things, which those results are forthcoming. So if we look at lead, we're talking about in the range of about 400 parts per billion. Probably the most alarming one is mercury, which is exceptionally high. Overall, these numbers place seabirds in kind of the upper echelon of chemical burdens for seabirds more broadly. And importantly, what we found is that the concentration of some of these elements, particularly things like chromium and silver, were elevated, significantly elevated in relation to the quantity of ingested plastic. So to us, that really does provide some reasonably strong evidence that if you consume plastic, the plastic is going to expose you to a range of toxic chemicals. So the next question was, can the exposure to this plastic not just change your body morphology and condition, not just expose you to chemical contaminants, but can it do other things physiologically? And if so, what? Again, we're dealing with wildlife that can't communicate how and where it's hurting or suffering. And so I kind of had to step back for a moment and think about how would you solve this kind of challenge. And one of the things that we often do when we feel unwell but we're unsure how to communicate that is we go to our local GP and we say, I don't feel well, but I'm not sure why. And your GP says, well, you know, as a starting point, we're just going to screen your blood. We're going to take a blood sample and we're going to test you for 5 or 10 things. And we're going to start from there, and that will maybe help us narrow it down. And so that's essentially how we treated the birds, is like a human. And so we took blood samples and we exposed them to something called a comprehensive avian panel. We sent them to a specialist veterinary pathology lab that specializes in diagnosing wildlife health conditions. And we tested the blood for around 20 blood parameters, including things like glucose, GLDH, which is an indicator of liver disease, white blood cell count, which reflects things like inflammation, and a whole other range of parameters. And what we found is that plastic was exhibited a positive relationship with a range of things, including the concentration of uric acid and amylase, both of which are associated with kidney function and also can be linked to things like fasting behavior, starvation, and also dehydration. So that kind of makes sense to us. If you've got lots of plastic in your body, you are again in poor condition, you haven't really been fed the right things, you're dehydrated, you're starving, all of these kinds of things. But most interesting to us was the results for cholesterol. The results really indicated to us, as you can see in this panel, an all or nothing response. Basically, the ingestion of even one piece of plastic appeared to kind of tip the birds over some kind of invisible threshold, where if you had plastic, you had high cholesterol, and if you didn't have plastic, you had relatively normal cholesterol. And so it kind of suggests that beyond some certain point, perhaps as little as one piece of plastic, adding additional plastic to your body burden maybe in some regards doesn't actually make things worse. It's literally a question of all or nothing. And that's interesting because we've never really been able to figure out, is one piece of plastic enough? Is that bad enough? So what have we learned from all of this and where to from here? Well, we've learned that the impacts of plastic are diverse, they're complex. In many cases, they're probably going to be quite long lasting. So when you have birds that are leaving the nest with reduced beak length, reduced overall body size, so they're quite stunted, they're going to have impaired survival in the longer term. And so what we want to do next is investigate, continue to investigate this and dig even deeper. And so this is where Jack comes in. Once again, we're looking at this from a human kind of perspective and saying, what kind of tools and techniques that have been used in human applications can we apply to these birds to learn a bit more and to dig a bit deeper. So at this point, I get the great pleasure of handing over to Jack, and he's going to explain what his expertise is and how it relates to wildlife studies of plastic.
Brilliant. Okay, so let me whip through here so we don't take over too much time. So what I was really concerned about is this gap. This is how I sort of came into the field, and it's this gap between innovation, application, and our understanding of the environmental or health consequences. One of my favorite examples of this big gap that we really need to be concerned about is the discovery of radium, the radioactive material. Then we started to apply it to glow-in-the-dark watches. And the factory workers were actually told to lick the brushes, the radium paint that generated the glow-in-the-dark watches, to paint the dial of the watch. But it wasn't until around about 54 years later that radium paint stopped being used, after the vast majority of workers got radiation poisoning or cancer. Now, this is just one example of this gap. There's loads of them: thalidomide, DDT, CFC, lead pigments, X-rays. When we discovered X-rays, rich people would get a daily X-ray to invigorate their health until we learned what it did. And now plastics. We're using plastics in everything. It's been applied to everything. Since its invention, it's proliferated massively. I've just had a baby and I've been out buying baby clothes. Virtually all baby clothes contain plastic. Nylon or polystyrene is their material. And are we right in the middle of this gap right now before we understand the massive environmental and health consequences these plastics are having? So that was the big question of my project, which I started in the University of Manchester before I came over to Australia. What are the health consequences of plastic innovation? And I was particularly interested in these microplastics, these really small plastic forms. Because we can't see them, they're not as obvious. I would know if there was a large chunk of plastic in me. You know, large chunks of plastic can cause obvious effects such as blockage of the gut. But perhaps these microplastics are having chronic sublethal effects throughout wildlife, throughout human health as well, and we're just missing them. But there's these massive effects going on. And so microplastics can go right down to very small scales, and it makes them particularly pernicious because they can actually infiltrate tissues within the body and cells. They can get into individual cells and cause inflammation in there. And that's where my expertise comes in. I'm pathologist and an immunologist. I really learned to understand what are the cellular mechanisms of tissue damage in response to stimuli like this. So there's two kinds of plastic. There's primary microplastics, which are made small. And examples are the fine fibers that you find in these polyethylene tea bags. And a study came out finding in 1 cup of tea, there's 11 billion microplastic particles being shed off this material. Another example is household dust. Our synthetic clothing is constantly shedding microfibers, so it's in our household dust. Around 33% of household dust was found to be of plastic origin. And this also happens when we wash our clothes. This just goes straight out into the ocean, these microplastics shedding off this clothing. The other kind is secondary microplastics, and these are macroplastics that break down in the environment because of UV and the oceanic environment. But also, this is a very interesting study that showed even digestive fragmentation. So organisms can eat plastic and then poop out smaller microplastics that can then go on to cause these effects. Now, during this, as Jen mentioned, during these processes, they're accumulating chemicals and their chemical structure is changing as well in response to the UV exposure. So my hypothesis was these materials cause chronic inflammation, which can have negative health consequences. So I'm going to give you, I know there's a lot of chemists out there and environmental biologists, environmental people out there, so I'm going to give a real quick breakdown of inflammation. Oh, I'm sorry. Okay, here we go. So this is tissue. This is a slice of tissue, and we can see the cells there. And we can see a blood vessel running through the tissue. And in blue there, that's an immune cell. And he's just surveying the tissue. I'm going to call her a she. She's just surveying the tissue. Now, should tissue damage occur, chemicals that are inside the cells will leak out. And they shouldn't be there. And so the immune cell, she has receptors that can detect these chemicals that are leaking out from dying cells. And in response, she will signal an inflammatory response. Another process is bacteria and other pathogens. They leak molecules that are detected on these receptors on the surface of the cell, sometimes inside the cell, to cause this response. So once we get the activation of the receptors, we get a release of a bunch of signaling molecules to cause inflammation. So inflammation, you'll get your blood vessels will dilate and they'll become leaky. Fluid will now rush into the tissue, and this is to allow more immune cells to come. So this is why when you roll your ankle, it starts to swell. It's filling up with fluid as well as cells. Now, all of this is a good thing because they're going to come in there, they're going to sterilize it, and they're going to clean it up. But here's the problem. If it's chronic, some of these processes can cause lots of damage. One really good example is if you see the round guy here spewing out little chemicals there, that's a neutrophil. And they actually produce a range of really toxic chemicals, including household bleach. That's right, your immune system produces bleach for the exact same reason we use bleach, to sterilize the area. But you can imagine that if you are chronically at a site of tissue producing bleach over and over again, you're going to cause huge damage throughout the tissue. And this is where inflammation can really become a problem. But you might be asking yourself, hold on a minute, how are these receptors going to detect plastics? A, plastics are insoluble, they're not really interacting with proteins, and B, they're a lot bigger than this receptor. So how is this going to work? How is there going to be the detection of the microplastics? And that's where this guy comes in. It's the most beautiful molecule I've ever seen. I imagine you're all questioning your research topics right now, thinking, I need to switch to this guy because that is the most beautiful protein ever. It's called the NLRP3 receptor. And what it does is it detects stress of the stomach of the cell. So cells eat stuff, they form a stomach, which is called a phagosome inside a cell. And if there is stress in that stomach, like something it really can't eat, this receptor detects that stress and activates. And when it activates, it causes the release of this inflammatory molecule called IL-1, which is the most inflammatory signaling molecule in the human body. So it is a very potent inflammatory activator if you've got something in your body that that the stomach of the cell can't digest. If a cell can't eat it, it's going to be very inflammatory. So here's some examples. Uric acid crystals cause massive, which is gout. Uric acid crystals that form in your big toe, that's gout. That's caused by IL-1 release in response to the cells eating those crystals and inducing inflammation. You can get silicosis of the lungs, which is when you breathe in sand. You can get massive inflammation in the lungs through the same process. Asbestos is caused by the exact same process. The cell eats it, it causes a massive inflammatory response, it dies. That asbestos is still there to be eaten again and cause a massive inflammatory response. Cholesterol crystals in your blood vessels, these are all caused by this IL-1 inflammation. And this is where I came from, Alzheimer's disease, where you actually have protein particulates in your brain causing chronic inflammation. So here's the question, does microplastics cause the stress? Does microplastics cause this inflammation? And it was about 2 in the morning, and I was supposed to be sleeping. I'm not a very good sleeper, but I was watching David Attenborough, and he was talking about microplastics about a couple of years ago. And he said, we know it's in bodies of animals and humans, but we don't know what it's doing. And I thought, there is no doubt in my mind, this is activating the NLRP3 receptor and causing inflammation. So I took a Swiss Army file to a tip box and got some microplastics, and I put it on some immune cells. And I didn't know what concentration I put it on. I could barely see any there. I really didn't expect anything. We call this the Friday afternoon experiment. And we got massive inflammation. Not quite as much as our positive control silica, but again, I put bucket loads of silica on. I had no idea how much plastic I put on. And importantly, we had immune cells that had genetically this receptor deleted. So it doesn't have this receptor, and we didn't see the inflammation. So we'd identified in one pretty simple experiment how microplastics can induce inflammation in the body of wildlife and humans. But we wanted to get more professional plastic than a Swiss Army knife. And so I wondered, where do you get good microplastics from? And so I teamed up with our engineering department, who actually makes parts for space equipment as well as airplanes. And they're able to finely grind down materials at micron scales. So this is the machine there, and we ground down our own microplastics. And this is my PhD student setting in the plastic there before it's ground down. And this is what it looks like. Now, it looks bigger than a microplastic, but they're actually statically clumped together. They're actually a lot smaller than that, but they just formed these macro clumps there. But this is nylon. So we applied nylon, polyethylene, and polystyrene to our cells, and we were quite surprised. Polystyrene was not inflammatory, whereas polyethylene and nylon, there in N and P, E, were inflammatory. And this again, when we put it on our cells that had this receptor deleted, was not inflammatory. So we knew how this was inducing inflammation. We looked at what were generated under scanning electron microscopes, and we noticed that the polystyrene was forming more circular fragments. So we wondered whether this relationship was due to structure and whether these shapes are more stressful on a cell than these shapes. So we wanted to see if this was biologically relevant, and this is a tough part of my job, but we did perform an animal experiment to see whether it would reduce inflammation and whether, because cell models are very basic, there's just one cell type there. So we injected the microplastics, a very small amount under the skin, and then we looked to see if there's inflammation going on. Now, this is the normal cell population you would see just under the skin of a mouse. And this is what we see here, massive influx of neutrophils. These are the guys that spray bleach. So this is massive damaging inflammation going on there, just in response to a very small amount of plastic. So next we tried a more realistic model, which is breathing it in, which is something we're doing right now, probably in your household dust. And each of these dots represents a cell, and we know what kind of immune cell it is based on where it's positioned in this graph. And we can see that in the placebo, there's a small amount of monocytes and neutrophils, which are immune cells. And in the nylon administration, we see a massive uptick of neutrophils and monocytes. So we see massive inflammation going on. Once again, this is a very similar response to what we would see with asbestos, which is very scary to me. But those were all primary microplastics. And what we're talking about with my collaboration with Jen is environmental microplastics, the microplastics that are in the ocean. So I actually asked Jen to provide me with microplastics that were collected from around where around Lord Howe Island, where our shearwater birds are, to see what kind of inflammatory properties do these environmental plastics have. Now we know, as Jen just showed, they have changes in chemical composition, chemical structure, UV, they collect heavy metals. So it's possible that this alters the inflammatory properties of the plastic. So we performed this experiment, we compared it to our most inflammatory naive plastic, we compared her environmental plastics. And we saw they were almost twice as inflammatory as the naive plastics, indicating that they've undergone changes in the environment that's made them more inflammatory. And it's still NLRP3 dependent, because when we put an inhibitor of this receptor, we block the inflammation. So now going forward, me and Jen are going to do a big deep dive, combining our skills of field research on these birds and my biomedical skills to see whether there's massive inflammation going on in these birds. And we're going to look at all the pathways. We're going to do large genome sequencing and RNA sequencing to see what genes are turned off and turned on in these birds to build a network of responses that occur during the plastics to fully understand the health impacts from plastic. I think an interesting finding from all of this was that some plastics were less inflammatory. Perhaps going forward, we can make recommendations about where the government should place their emphasis on plastic cleanup and encourage people to use different, safer, less biologically active forms of plastic. And just to note, Jen already showed this information, but you get this elevated cholesterol and elevated uric acid levels. We see these kinds of chemical signatures in the blood of patients that have undergone chronic inflammation. These are symptoms of chronic inflammation. So it's highly likely that these plastics in these birds are causing chronic inflammation, which can lead to massive health outcomes throughout every biological system in that bird. So thank you very much. And Jen will unmute as well to say thank you, I imagine, for listening. And we're all happy to answer questions. And hopefully I got through that roughly on time.